Good afternoon.
To one and all. Good afternoon to one and all, and good afternoon to today's resource person, Dr. MP Venkatesh sir. Now we will start the second session of day 4 by Dr. MP Venkatesh sir session. Now I would like to invite Dr. MP Venkatesh sir to start the second session. Thank you.
Good afternoon, one and all. You all know me since I gave you the first presentation a couple of days back. I'll be moving to the talk of today's discussion, giving you the basics of the different phases of clinical trials. Let us move into the talk. Clinical trials happens to be one of the very important aspects of a drug development cycle, which involves testing of the newly developed drugs or drug products in human beings. In broad, the term clinical refers to the aspects or the activity which we do it in human beings. During the process of a drug development, the investigational medicine, that means the drug which has been undergone the process of new drug discovery and development, after it has done through sufficient amount of laboratory examinations and development, it has to be tested in the human beings before the regulatory approval is given. To do this, we do the trials in human beings, which is said to be an systematic study of the novel new drug delivery system in the human beings. So the trials are done in a series of steps, which we call it as phases. To describe, there are 4 different phases of clinical trials. We say phase 1, 2, 3, and 4. This is a nutshell or the diagrammatic aspect of what is a clinical trial all about. We have seen the activity where a preclinical studies will be done, that is the studies which will be done in an animal model to prove the safety, efficacy, dose ranging studies, acute studies, and even the the long-term studies along with the toxicity studies will be done before it is actually applied as an investigational new drug, which will grant the application for conduct of the clinical trials in human beings. That means all these aspects are done as a part of your preclinical studies. Once it is done, the company or the applicant have to submit an investigational new drug application. The regulatory authority will review the investigational new drug application. If they found it good, then they are going to approve for the conduct of clinical trials. The first part, what we do is phase 1 studies, which is a part of the initial clinical studies. If it is done with, they move across phase 2, phase 3. At the end of phase 3 clinical trials, the data which is generated as a part of your clinical trials of phase 1, 2, and 3 is combined and submitted to the regulatory authority as new drug application, NDA. So the regulatory authority, if they are satisfied or if they see the fulfillment of the requirement of the data, then they're going to approve the new drug product for use in the larger population. So once the drug enters into the market as an approved drug product, then it is available for general public to take this medication. And parallelly, there is starting of phase 4 studies, which is called as post-approval marketing studies or post-marketing studies. If the drug is successfully passing the different phases of the clinical trials, which will be usually approved by the national regulatory authorities, which we call it as NRA, for use in the general population. The phase 4 studies are called as post-approval studies or post-approval marketing studies or post-marketing surveillance studies. So it is a requirement that all the pharmaceutical companies have to start the clinical trials on the drug product which they have developed. They have to conduct extensive preclinical studies before the actual clinical studies will be taken part. Let us see by one phase by another phase. The first one is phase 0 trial. Now in the previous slide, I said it is phase 1, which we have never mentioned about phase 0. Phase 0 is an optional clinical trial, which will be done for certain products and it is not a compulsory trial. Generally, they are called as microdosing studies. This is a new idea of the US FDA, that is United States Food and Drug Administration, where phase 0 studies is not a mandatory clinical trial, similar to what you have seen with phase 2, 1, 2, and 3. The idea is if you are able to give the dose at 1% of the therapeutic dose to a few volunteers, they will try to evaluate what is the safety and efficacy of the drug, so that whether a therapeutic effect has been seen or not. To make it simple, we say if the dose of the drug is able to, which we thought of like 100 mg. So in the phase 0 clinical trial, what we do is we try to administer 1 mg of the drug to a very less number of people. So with this, what you do, we try to evaluate whether there is any adverse events or if the safety of the drug is good or not. If it is fine, we'll go with phase 1 trials. You may ask a question, why if you administer at a very low dose, what do you get? There are many instances where even the therapeutic dose, which we have calculated at the end of the preclinical studies, sometimes it could be fatal. To prevent such misuse, the misadventure of the drug, what they generally do is, in very rare condition, if you feel that the drug is able to produce any serious adverse events, even in the low dose, they try to evaluate that as a part of your phase 0 microdosing studies. It will even show whether a drug's pharmacokinetic and pharmacodynamic principles will warrant and continued exploration, meaning that if you continue to explore the drug at a very low dose, then by increasing the dose to a therapeutic dose, we get more advantageous in the method of gathering the data. Generally, at the end of the phase 0 trials, we can say either you go or not to go. That means you can move into phase 1 studies, or you can stop not doing any further studies rather than relying upon the animal data. So what is the entire basis of doing the clinical trial? The entire basis of doing a clinical trial is just the animal data, which has been done in different animal models, at least 2 rodent models followed by a mammalian model. So this will give you, the phase 0 will give you an idea whether the drug tested for the first time in humans is able to produce the efficacy, and more important, whether it is safe for administration or not. And as I said, that is only an optional studies. It is not mandatory. The first clinical trial, what is being done in humans, is called as phase 1 trial. This is called as first in human studies or human pharmacological studies. The tagline of this particular phase is, does the drug really work? Is it safe? How does it behave in humans? So this is the entire idea. What is all about this phase 1? First, whether the drug will work, whether it is safe for administration. If I take this drug, what will happen in my body? The aim of this phase 1 studies is to ascertain the safety of the drug, whether if I administer this drug into the humans, it is safe or not. The second one, once you are able to see the safety of it, we try to identify the range of dose which has been tolerated by the human beings. Like many a times you say, the drug will be taken in 10 to 30 mg per day in divided doses. That means 10 mg is the minimum therapeutic dose, and it can be taken up to 30 mg. That is how you're going to get the information. So this is done as a part of your phase 1 clinical trials. During this, you are going to study the ADME characteristics: absorption, distribution, metabolism, and excretion. For deriving all these above information, the population or the study subjects, that is the human volunteers, we take about 10 to 20 normal, healthy human volunteers. And to do these activities, we conduct some 8 or 10 tests in the patient population to check what is this actual activity. To do this, the cost incurred is 10 to 20 million US dollars. So it is not a small amount of money. It is a very huge thing. Even though the number of patients is only 10 to 20, the expense is very high. After an experimental drug or a treatment has been tested in the lab or in the animals after in vitro or the preclinical studies, generally it enters phase 1 trial. In this trial, a new drug or a treatment is tested in a small group of people for the first time. That means till now, this drug has never been evaluated in human beings. First, they will evaluate the safety, determine what is a safe dosage range, and if there is any side effects, they are going to check all of them. The goal is to prove that the new drug or the treatment which has been proven to be safe
Sorry, sir.
mute your mic.
Can you Mohammed? Sir, just give me 2 minutes, sir. I think there is a technical issue.
Yeah, ma'am. The goal of this phase is to prove that the new drug or the treatment which was proven to be safe in the use of animals may also be given to humans without any much problems associated with it. So what is a dose range? So what actually we do in this is the trials of phase 1 will help to determine what is the best way to give the drug, whether we can administer the drug by oral route or by intravenous administration. In phase 1 clinical trials, the dose of the investigational drug is gradually increased to check what is the optimum or the safe dose. The process is called as dose escalation. In the initial cases, I'll give you less dose, maybe 5 mg per tablet. Later, I'll increase to 10 mg, 15 mg, or 20 mg till I see a visible or a noticeable side effect. When a small dose of the drug is given, if there is no side effect, then the next group is given higher doses of the drug until the doctor is able to determine what is the optimal dose with less side effects. The phase 1 clinical trial will generally go up to several months, and it will involve a small number of people, usually 10, but not more than 20. The most phase 1 clinical trials are initiated using male fit volunteers or male healthy volunteers who may be recruited by various teaching institutions or the medical schools or even the hospital-based data. So within phase 1, you have 3 kinds. One is the ascending dose studies, the single ascending dose studies, multiple ascending dose studies, and even what is the effect of food on the ADME characteristics of the drug. The first one is single ascending dose study. In this thing, as the name specifies, you're going to give a single dose in an ascending order. The process involves where you take a small group of people who will be given a single dose of drug and who will be observed for a period of time. If the tolerability of the drug is good, and even if the pharmacokinetic data and the other data is good, then if it is within the predicted safe values, then the dose of the drug is increased in the new group of subjects. This process will continue till a maximum tolerated dose is reached. If you can see the chart here. In the first cohort, we'll be giving 5 mg. If it is tolerable or if the levels are found to be good, then in the second week, we give to Cohort 2, 10 mg. Similarly, I start increasing the dose 20 mg, 50 mg, or even 100 mg to see whether the dose administered is tolerable with good, safe pharmacokinetic data. The second type of phase 1 trials, what we do is multiple ascending dose studies, where a group of subjects will receive multiple low doses of the drug and it is observed for a period of time. During this observation period, we collect the blood samples, other body fluids, and they will be analyzed for various parameters. During this, we should be able to understand the kinetics and dynamics of the drug. And the entire process is depicted in this table. In cohort 1, in the first week, I'll give 20 mg. I see the tolerability and acceptability and the safe levels. In the second week, if it is tolerable, I start the double dose of it or increase the dose of this to the cohort 2 as 40 mg. Apart from this or along with this, to the same group, I administer the increased dose as 40 mg. So now this is something like your single ascending dose studies. Along with this, to the same group, I start administering the higher doses basing upon the tolerability. So this will give an additional information whether the drug has any accumulation potential or the toxicity related activities can be found out by giving the drug in increased order to the same group. The third one is the food effect, as we can understand whether the food does have any impact on the absorption of the drug or vice versa. If I take the drug after the consumption of food, whether it is going to affect the process of dynamics and kinetics. They do in two ways. One, they do it in fasted state. That means the patient has not taken any food for the last 8 to 10 hours, then the drug is administered. And after the food has been taken, the drug is administered. That means before or without the consumption of food and after the consumption of food, what is the effect of food on the drug absorption and kinetic process. So this is one of the method where they do with the dose escalation studies in phase 1, that is called 3+3 design. For the first 3 patients, they administer 10 mg. So next 3 patients 20 mg and next 3 patients is 30 mg. Once the dose is determined, they will extend the number of people to 10 or 20. So they wanted to do in a small number of people first, then they're going to expand. This is generally called as 3+3 design. The phase 1 clinical trials, if the results are successful and if it is good enough, they move into phase 2 clinical studies or phase 2 trials called as therapeutic exploratory studies. The important aspect is whether your drug is really efficacious in the patients or not. The aim is to have a first look about the efficacy. Till now, we have only analyzed the safety, not the efficacy part. Now, you're going to see whether the drug is efficacious or not. So the design includes the placebo control design. That means you're going to use a placebo followed by the process or the trial. What we do is randomized and it is double-blind studies. It is done at selected medical research centers where the study population is about 200 to 300 patients who are really suffering from this disease. That means you're not going to use healthy human volunteers. You are going to use or involve or enroll the patients who are suffering from this particular illness. The scope of this is it will take almost 2 years and the cost is about 20 to 40 million US dollars. At the end of the studies, you can understand whether you need to go to phase 3 studies or you want to stop the entire process of clinical trial here. In the phase 2 clinical trial, we'll understand whether the initial safety of the study or the drug has been confirmed from phase 1. If it is yes, then we go with phase 2 trials. The experimental drug or the treatment is now given to a large number of people. There we gave to 110 to 20. Now here it is exposed to 100 to 300 people to check whether it is effective or not. So when you do this, you gather additional information regarding the safe dose range of the drug. Generally, the phase 2 studies is divided into phase 2A and 2B. The phase 2A is done to assess what is the dosing requirement. That means how much drug should be given to get therapeutic benefit. Phase 2B will be designed to study the efficacy of the drug, how well the drug will work in the prescribed doses. As I said, we enroll patients for this particular phase 2 trials. In the phase 3 trial, that is phase 2 is called as therapeutic exploratory phase, where we are going to explore the possibility or the dose range of the drug. In phase 3 trials, it is therapeutic confirmatory studies. Whatever we have done in phase 2, the results, whatever we have got, that will be confirmed in the phase 3 trials. It is done to establish the safety and efficacy in a large patient population, about 2,000 to 3,000 patients, which is done at a multiple or major hospitals. And it is usually multinational clinical trials where it is done across multiple countries at the same time. This is the pivotal studies which is going to decide whether the drug will have the chance to enter the market or not. The design is randomized, where we use controlled method of conducting the clinical study, and it is done in a double-blind way. The term double-blind means neither the patient nor the doctor or the healthcare worker will know what has been administered to the patient. Either it is placebo or the comparator or the test drug. Generally, it will take up to 3 years, or 100 to 150 million US dollars will be spent on the conduct of phase 3 trials. The problem statement or the statement here is, does the new treatment work better than the standard treatment? That means you have a standard treatment already, which is being used for the treatment of a particular disease condition. So against which, if you want to find a new drug, they're going to say this. They're going to monitor the effectiveness, monitor the side effects, compared to commonly known treatment which is already there or which is already being in use. And this will even allow us to gather the information about the drug which can be used safely or not. The phase 3 clinical trials are randomized, meaning that the patients who are enrolled for this study will be grouped into multiple groups, like placebo group, comparator group, control group, or the test group, where the people who have been grouped is done in a non-orderly way. That means there is a randomization technique by which the system or the computer or a software, basing upon the parameters of the drug or parameters of the patient, they will be grouped into multiple groups for the purpose of conduct of a clinical trial. Although phase 3 clinical trials will focus within a specific disease, they generally include people of various ages, generally more than 18 up to 45 or 50. The ethnicities of people will be different. Both male and female will be enrolled into the study so that all other parameters are considered for the process. Since the number of people enrolled into the study is several thousands, it will take 5 to 7 years to complete phase 3 clinical trial. Because of its longer duration, phase 3 is said to be one of the most expensive, time-consuming, and very difficult trial in case to design and even conduct the clinical trials, especially if you're talking of medical conditions which are very prolonged in nature, which require treatment for a very long time. Once the drug has been approved or proven satisfactory after the conduct of phase 3 trials, usually the information, whatever we have got till date, will be now compiled, compiled as a dossier, and this will be a part of your regulatory submission. So this document will contain the methods and results of the human studies, animal studies, how the drug is being manufactured, how it is formulated, and even conducting of the shelf life, and that thereby you'll be doing the stability studies of the drug. The data which has been compiled as a dossier is now submitted to the regulatory authorities of different countries. The regulatory authorities will review the data right from the development of your molecule till the completion of the clinical trials. If the data is found to be satisfactory and meeting the requirements, they will approve the drug for treatment, and then the drug is given a go-ahead or a green signal to enter the market. So the drug will enter the market, and now this will be used by a very large pool of patients. So in the clinical trial phase 3, we use only 2,000 to 3,000 people. But in phase 4 trial, or once the drug comes into the market, the number of people using the drug will have no limit. It can be 10,000, or it can be 1 lakh, or it can be 10 lakhs. The number of people using, or the patients using the drug, is very high. As a part of the mandatory requirement, the manufacturer or the applicant have to do phase 4 clinical trials, which is called as post-marketing surveillance studies. This is generally done to check, or it is done to review or to survey, do the surveillance of the drug or the treatment, whether it can be used for the actual purpose, or whether you can use it for any other problems or not. It is designed to detect any of the rare or long-term adverse events of a drug in a very large population. So phase 4 clinical trials will continue for multiple years before you can establish the safety profile of it. If you find any new adverse events or side effects, etc., that will contribute to the labeling requirements or precautions or any of these aspects relating to the drug molecule. That is how you conduct the different phases of the clinical trials. So this completes the first part of the basics, what is all about the different clinical trials or different phases of clinical trials. If you have any questions in this, you can feel free to discuss on that. Vijay ma'am?
Yes, please.
The first part of discussion is over. If there are any questions, I'll take it. I have a second presentation. That is a clinical trial protocol.
Okay, sir.
If you want me to complete with that and then take questions, is also okay.
So students are requested to post their doubts if they have in the chat box. So sir will check and clarify the doubts. So,
or should I complete the second presentation then?
Or otherwise, you can unmute yourself and ask the questions directly if you have any queries. Otherwise, we will continue to the next session. Okay, sir, please consider that as they don't have any queries, please continue the next session, sir.
Okay. Next part is the clinical trial protocol. So for the conduct of or to carry out clinical trials, this is one of the very important document which will give you the complete details about how an clinical trial have to be conducted. So this is a mandatory document for all the phases of clinical trials, without which you cannot even start or even think of doing any clinical trials within the requirement. There are different trial documents. That means the documents which are required for the conduct of a clinical trial. The first and foremost document is the protocol, or which we talk of clinical trial protocol. The second one is the investigator's brochure. We call it as IB. You have informed consent document, case report forms, in short, we call it as CRF, the clinical study reports and summaries, contracts and agreements, and even diary cards. There are many other numerous documents which have to be done before the actual start of the clinical trial. This is a very important statement. The beginning is the most important part of the work. That means how do you start something is everything. So if you're able to do things properly, then most of your work is done. Bigger question, what is a protocol? The protocol is a document that will provide the background and the framework for the planned study and describe how it can be implemented. Meaning that it is a document. That means it is a written document that will give us what is the background of the conducting of this study, the framework, how exactly this have to be done, and it will even describe what are the steps of implementation also. Generally, protocols are a very detailed document which is written in a very thorough manner, giving all the information, and it is compiled by a clinical trial sponsor personnel. The term sponsor is nothing but a company who is investing on the conducting of a clinical trial. The individual investigator, the term investigator refers to be the doctors or the clinicians who is part of the study, who will be actually doing the intervention in the human patients. It can be scientists of different origins, or it can be combination of any of these people. In the United States, the trial sponsor or the company will submit the final protocol to the US FDA as a part of your IND application, that is investigational new drug application, or IDE application, that is investigational device exemption. This is generally done for your medical devices. Before the conduct of an clinical trials, the FDA must approve the protocol so that the investigator can investigate the aspect in their clinical trial sites. Sometimes we may not require a protocol for the conducting of certain trials. Example, the protocols for clinical investigation of marketed drugs, that is phase 4 clinical trials. Some trials where there is only observation. That means you don't give any drug for the patient, you don't inject anything, nor you provide anything. For such kind of studies, you may not require a clinical trial protocol, but either way, you need to provide, or you need to take the approval from Institutional Review Board. So Institutional Review Board is nothing but the authority who will be reviewing the protocol, that the clinical trial protocol of a clinical trial, and this is the body which is going to give approval and even the monitor the progress of the clinical trial. And this is a very important body, that is Institutional Review Board, which is having the regulatory approval or regulatory importance, because for everything the clinical trial, it has to pass through IRB, later to the regulatory authority. Let us look into what are the different components of the protocol. This is the TOC, that is table of contents. What you have for a protocol is introduction, background, rationale, objectives, if they have done any previous studies, followed by the endpoints, what is the trial design, how the subjects are selected, randomized, the treatment plan, how the assessments are made, the different aspects of the drug, how the dosing is being done, how do you store the drug, accountability, data collection, and many more. Let us see the important aspects of the protocol. One of the very important thing is giving the background of the study. So before you start doing any activity, you have to see what is the background. So why you require to do a clinical trial? So that means you have to provide what are the requirements, what are the results, what are the studies which was done earlier in this connection, and what is the safety and efficacy information you have obtained from the previous studies. Basing on the background, you need to provide a rationale why this trial is being conducted, and it should be even consistent with the background information which is provided by the previous section. Study organization will talk of how the structure of the study is organized, which is based upon the requirement of the protocol, the financial needs, and even the logistic issues. The term logistic refers to the arrangements which is made for the men, material, and goods from one place to another place, how the movement will happen, storage, transportation, everything have to be given over here. The endpoints is nothing but these are the points on which you can say whether an objective has been achieved or not. To say, like if your drug is for antihypertensive agent, so how do you know it is? What is the endpoint of it? If the blood pressure is reduced from 160 to 140 or 120 kind, something like it, that means if you get a set value, then it is said to be reaching the endpoints in general. So endpoints are measures which is believed to quantify. That is why I said you need to have a measurement in units or numericals. Quantify the potential effect of a treatment or therapy under the clinical trial. In addition to the clinical endpoints, sometimes even the quality of life and economic factors can also be considered as endpoints. So what is quality of life? To quote an example, where we have a person suffering from rheumatoid arthritis. So the person's joint has given him or given the patient a very huge pain or very severe pain where he cannot move his limbs. Your treatment is made such a way that the pain in the limbs is reduced, where he can walk, sit, and stand comfortably. That means the quality of the life of the person has been improvised. So that is said to be an endpoint. If you talk of a clinical endpoint, the clinical endpoint should be relevant and it should be easy to interpret. It should be visible clinically. That means it should be apparent whenever I look into the endpoints or if I look into the parameters, I should be able to determine, yes, if this is happening. And it should be easy to identify. And if I make any changes in the treatment, the endpoint results have to be varied. That means it should be sensitive to the treatment differences. So when you look into all these things, we have to write the protocol in a proper way. So this is nothing but protocol is a written document. So within the document, the first and foremost question is, what is the aim? That means what is the question in mind for which you are doing a clinical trial? And what is the expected outcome? Whether the new treatment is better than the old treatment, whether the new drug has less side effects, whether the treatment under question is having better acceptability, all of them. In some cases, they will even go for an intervention or administration or a particular activity. If you're doing any intervention, for whom are you going to do, when, and for how long? And in the protocol, we have to define what is the number of participants or the patients or the subjects who will be enrolled. Say 10 patients, 25 patients, 80 patients. So we have to tell them, and we have to optimize what is the risk and benefit ratio. In the background information, you do a lot of literature review. Within the literature review, you have to address all these things. What is the safety assessment of your drug? Whether there was a lacuna of the therapeutic requirements, or the delivery system, what I'm doing here is new. Example, you had tablet earlier. Now I'm getting an injectable dosage form. So that is a new drug delivery system. New dosage schedule. Earlier I was taking thrice a day. Now I need to take only once a day. Or the drug was earlier used for antihypertensive thing. Now I'm using for prevention of hair fall. So it is in exploring the new indication. Sometimes you need to just monitor the therapeutic advantages of the drug for safety efficacy, or I have got everything, but only regulatory filing is pending. The aim or the question of the clinical trial, whether it is properly defined in the trial protocol or not, whether the results will demonstrate the new things compared to the existing knowledge, whether the ongoing studies will address the same aim. That means you have to do a thorough research saying that if any person in the world, in any other country, are doing a similar study or not. If they're doing a similar study, then there is no need of you to carry out the same studies in a different country. So that is how you can do. And the overall advantage is whether the trial will provide any new knowledge or advanced patient care to the people if this is successful. Generally, the aim of the study is one or two. That means you don't have multiple aims doing in a single study. Example, you compare the efficacy between two drugs, or to check the effect of something on the blood variation, or to check the efficacy in respect to age or sex or any other associated conditions. That means you need to have a simple or a simpler aim with one or two objectives. If you have multiple aims, that means the study design becomes very complex and it will become very difficult to execute the protocol. Generally, they will not have more than two aims in a single clinical trial. The endpoints can be either the primary endpoints or the secondary endpoints, which we have discussed. The primary endpoints are the one where you can see the clinical benefits. The secondary one can be related to the safety and the tolerability of the drug. Now, entire clinical trial, whenever you're doing, it is based upon hypothesis, meaning that my trial is able to give a better treatment modality, it is better acceptable, or it is better than the existing drug. It is all about the hypothetical or hypothesis which a drug manufacturer will have the question. The hypothesis is generally based upon the aim of the study results. Generally, if the hypothesis is said where the existing drug or the new drug, if they have the same efficacy, then we call them as null hypothesis. That is, both the treatments are equal. Treatment A and treatment B are same. That means there is no greater difference between the new treatment and the existing treatment. The hypothesis can be alternative hypothesis. First, you need to prove that A or treatment A is not equal to treatment B. That means it can be two-tailed or two-sided. That is the outcome of the study. Now, if you say A is not same as B, then you can have two things. Either A is better than B or B is better than A. If this is the case, then we say it is one-tailed or one-sided outcome of the study. The very important part of the clinical trial protocol or the execution of a clinical trial protocol is patient selection. That means you need to enroll the people or the patient for the conduct of the clinical trial. So the patients who are enrolled as a part of your clinical trials should have certain criteria. Not everyone can be involved or enrolled into the study. There are two sets of criteria. One is called as inclusion criteria, which will tell whom could be enrolled into the study. The other one is exclusion criteria who should not be involved or enrolled into the study. That means you need to have a precise clarity or the precise data against which you need to either include or exclude the patients. Example, to give, you need to have a precise diagnostic limits or the criteria of the disease to be treated. Example, if the drug is related to treating an hypertension, then who should be enrolled into the study? Example, the person should have a systolic blood pressure of more than 100 mm of mercury in the sitting position. The person should have glycosylated hemoglobin more than 7.5%. So they have to be very clear what is your requirement. If your drug is, it is for the treatment of any heart-related diseases like myocardial infarction, they say the ECG changes should be like this, the rise in the CPK enzyme levels, the age limits, the sex, either male or female can be enclosed or enrolled, or if the person has any associated disorders along with this problem, if they have any other diseases, all of them, whether they're taking any other treatments for any other associated diseases or disorders, everything should be part of your patient selection that is in the inclusion criteria. Sometimes before you enroll the patients into the process or after you enroll, there is something called as baseline establishment. During this period, what they do is they try to get rid of the remnants of any drug which is used by the person. Example, a person underwent a certain antibiotic treatment for a particular infection in the body. They know the drug is some antibiotic like amoxicillin or chloramphenicol or something like that. So they know what is the half-life of the drug, when was the last dose of the drug which is taken, and what is the time taken for the complete expulsion of the drug from the body. So if that is the case, they have to wait till that period, try to see that all the drug remnants or the accumulation of the drug is completely removed from the body. Then only they start with the clinical trial process. The opposite to that is the exclusion criteria. This criteria will define who should not be enrolled into the study. That means if the person is falling into these criteria, then I will not allow them to participate in the clinical trial. First and foremost, which is very common for most of the drugs, is pregnancy. The women who are pregnant is blanket ban. That means they should not be taken into the study unless until the drug is proven to be safe for the fetus. But generally, they don't take or enroll any pregnant woman. The second one is lactating mothers. So once the baby is delivered, the mother will be providing the breast milk to the baby. In that cases, the new drugs should not be administered to those conditions, because there may be a chance that the new drug may be excreted via the breast milk, and that can go to the baby and it will harm the health of the baby. So in that, this is a very clear blanket ban for pregnant and lactating women. They should not be involved or enrolled into the clinical trials unless until they are required and it has been proven that it is not been a worrisome factor. Apart from this, if the person or the patient is associated with any other conditions or the treatments which will interfere with the existing protocol, then they are not enrolled. There are some contraindications for the study treatment. If the person has done all of them, then they are not enrolled. If they have any relative contraindications, which is listed in the above thing, is not welcome. And last one, the patient says he cannot come back for follow-up, meaning that after the administration of the drug, we need the patients to come into the clinical trial site over a period of time for monitoring the requirements of blood or urine or any other samples within the patient. So if the person is unable to come to the clinical trial site at regular intervals, then those persons will not be generally enclosed or enrolled into the study. So how do you evaluate the efficacy of it? So the criteria of evaluation can be very important. That is, you need to do the efficacy comparison between the two treatments, define what is the endpoint, what is the mortality rate. Mortality is death rate. Whether the person is able to complete the study treatment duration, whether all the targeted values like blood pressure, glycemia, or any other things can be achieved or not. So all of them is a part of your endpoint. During this process, you need to define the method or the condition or the techniques by which you do a particular activity. Example, if you're taking a blood pressure reading, whether the person is in a lying position, that is sleeping position, sitting position, or standing position, because your posture changes, the blood pressure will also change. If the person has come after running and sitting, if you take the blood pressure, it will be more. If the person walks to the area, immediately if you take, it is somewhat less compared to running. And if a person is stationary for a certain period of time, and if you take the blood pressure, then it will be altogether different. So your activity, your posture, everything will affect the blood pressure. So you should be able to understand what is the requirement or what is the technique at which a particular activity have to be done. Apart from this, you need to develop an analytical method which is validated for estimation of the principles or the drug or the metabolite. Sometimes you may require to collect even urine samples or blood samples or any other samples from the body which is required for evaluation or estimation. As a part of your good laboratory practices, all your equipments, instruments, everything have to be calibrated every day before the actual usage of it. You need to schedule various tests. What are the tests to be done, when it has to be done, whether the number of tests to be done per day, the time of collection of a sample, analysis, everything have to be done. The frequency of sampling. So this is a very important part and ethical part which talks of how, when, and what are the considerations for sampling. Example, if I take a blood sample, if I say I'll take blood sample once a day, it's good enough. If I take 4 times a day, that is bit annoying. If I take 10 times blood sample per day, that is irritating, right? So you should be able to understand what is the practicality of it, whether it is ethical for certain actions or collection of a sample or any other activity per day. So whenever I do this testing, so who will measure the parameters? Whether the investigator or the clinician will do, or a rater who have been trained for that particular activity will do, or if a rater is being doing, whether the same rater will be doing multiple times for the same qualitative assessment. Anytime when it comes to the word like qualitative, it is all basis of a judgment will be there by the person. There is chances of bias. So it is always better you have an quantitative assessment of a particular activity or the parameter. Generally, if there are any issues, we will be having 2 independent observers who will be evaluating the same report and see whether they have any differences between them. If there is any difference, we apply certain statistical techniques for analysis. So data collection, again, collection of the data for a particular activity across the demography is a very important measurement. So this will be helpful for checking the comparability between the groups. It can be demographic characteristics, that means the population who have been spread across multiple places, the physiological characteristics of the patient, whether the signs and symptoms, when it has been onset, what is the time taken for onset of all of them, the other related factors, the concomitant illness and therapy which is taken by the person or which is seen in the patient during the study. For data collection, is there enough room for notifying each result? That means when you talk of the case report forms or any other things where you need to have enough amount of place to capture the data and to verify the results and correlate the results every time. If it is not there, then there is no meaning of collecting additional data. Moreover, are you collecting the data in the logical order? If there is any advantages and disadvantages in the order of data collection is also better. So all these things are a part of minute detailing of the clinical trials. Sometimes we may see certain adverse events. If the drug is tolerable, then you don't see any adverse events. If it is intolerable, then you have adverse events. That means the side effects, what you see, is said to be a part of your non-tolerability or intolerability of the drug. Generally, if there is any intolerability of the drug, it will be narrated by the patient to the caretaker. So whenever you are asking, whenever you're coming across any adverse event reporting, usually the caretaker will ask questions. The questions can be either open-ended questions or closed-end questions. Example, what is an open-ended question? Meaning that, okay, you said that you are non-tolerable to the drug. Tell me what happened to you. This is an open-ended question. So they can tell what is the description of it, how the treatment happened, what is the outcome of the study. All of them, the patient will narrate to the caretaker. What is a closed-ended question? They'll ask, okay, what happened to you? What time? Right? Or which place are you from? When did you take the drug? It is all a closed-ended question because you get a definitive answer. So when did you take the drug? Which packet? What ml? So everything is a closed-ended question. Generally, a mix of open-ended question and a closed-ended question will be asked by the caretaker so that they should be able to provide additional information or the complete information. Sometimes they even monitor for the safety of the drug, where you need to justify the results of the lab evaluation, where the lab evaluation can be a part of your routine studies, which is done as a part of your new drug, and it is directed by the various pharmacological efforts. Further, the laboratory evaluation will be done by collecting the samples from the patient. So there is a requirement what tests have to be done before and after the end of the study, or the central lab or the site where it has to be done. And there will be a standard symptom checklist against which the person has to check whether the symptoms are because of the drug or the dosage form or not. The compliance requirements for all these activities, you need to comply with the requirements. So compliance is the method by which you should be knowing whether the drug related aspects will be met or it is not met. So generally, if it is for a treatment, that means the compliance requirements happens after the treatment. So you need to define what is the procedure for the treatment and what medication is used in what doses, in what duration of time is to be given. If there is any compliance to be met in a non-drug treatment, that means you're not administering any drug to the patient, but the patient have to fulfill all of them. Meaning that he should not take high protein meal, or he should not take any high fat diet. He should only be on high fiber diet. That is all dietary restrictions. And the exercise regimen. Example, the person has to jog for 1 kilometer. He has to do these exercises. So all of them is a part of your requirement for compliance. So once you have said the compliance to be made, you need to check whether the person or the patient is complying to the requirements or not. So how do you know it? You need to have a questionnaire where you can ask what diet did you eat, when did you eat, what was there in your plate, what was given in your box. So all of them, once you are able to, if the person is able to define them, that means the person has taken that. Similarly, for the tablet count, example, the number of tablets given to the patient, how much strips or blisters or containers they have used. If they have unused, they'll tell, so these many tablets are not taken. So that is how you can measure. Sometimes, if you know the drug is excreted by a certain route and the drug excretion will have a markers. Example, if I take this drug, my feces will be brownish in color or black in color, or my urine will be turning red. So that something like this, which is a peculiar observation which a person can make, is also presence of a marker of a drug, which can be easily understood by the caretaker or the clinician, so that we say the patient has taken the drug properly. So similarly, we have other methods of finding the drug or metabolite in the biological fluid like blood or urine, etc. You can have a recording device which is incorporated into packaging. That means the packaging device is such a way that if I open the blister, it will note it down. So 16th of November, 3:10 p.m., the packet has been opened. That means the person has taken the drug at that particular time. So you have even memory aids for this particular things. The experimental design talks of the prospective and randomized. That means the term prospective refers to this is a study which is aimed to provide a benefit. Randomization, where the number of patients is grouped into different groups basing upon a definitive criteria. Sometimes we match the subjects. That means if 4 people are from Bangalore, if I have 2 groups, so I put 2 people in one group and 2 people in the another group. That means I'm matching the demography. Similarly, the severity of the disease. The factorial design is something that where you need to use 2 or more factors to be used as a part of your randomization technique. The term crossover is a very, very important aspect in the clinical trial, where if 2 treatments are given, if one treatment is placebo, the second treatment is test. After a period of time, once the treatment is over, the first group which was given placebo is now given the test drug, and the group which was given the test drug is now given a placebo. That means each group is evaluated for both the factors. So this is how the crossover design is done. And we have techniques called as Latin square design, which is used for randomization. This is the factorial design where you have 4 factors: exercise, no exercise, drug, and placebo. So I can give one group, drug is given and they have to do exercise. The other group, drug is given, they have not to do exercise. So similarly, you have 4 twos, that means 4 square, you can have 16 different factors which can be considered for this. Rather than 2 by 2, you have 3 by 3 square, 9 groups. So you have blood pressure, low, intermediate, high, blood lipids, low, intermediate, high. So you can have a combination like this. So 3 square or 3 cube design, you have 27 possibilities over here. So other stratification like male and female, age less than 45 and more than 45. Again, the groupings and the stratification can be made. This is the Latin square where you have subject 1, 2, 3, grouping 1, 2, 3. So you can have ABC, the 3 square, 3 cube design. The same thing is mirror image. That means for the right hand part, if you can fold and place it on, it will be again closing back onto the same thing. So this is called as the mirror imaging of a Latin square. So you can use it for different treatments. Now, how the treatment is given for the patients who are allotted to different groups. Generally, the allotment of treatment can be based upon packaging number, where you have sealed by opening the sealed envelopes containing the randomization codes. By even randomization can be done by telephone or even the data entry. The blinding of the study treatment. So if I make 2 groups, placebo group and active group, both of them have to be given a similar looking treatment cover or a tablet or anything like it. If a person is able to identify which is an active and which is a placebo, that means the blinding is not properly made. That means if the code can be easily broken, that means you have to change the design. As a part of your normal clinical trial, what we do, we generally use placebo for one of the groups, meaning that a placebo is a drug which is looking same exact as of the treatment drug, but it does not have the drug in it or the active in it. Example, if you have cisapride tablets, or you can consider paracetamol tablets, the test group will have 500 mg of paracetamol in each tablet, whereas placebo will look exactly the same of the drug containing tablet, but there is no drug in it. Now, why should we use placebo? If I use placebo, then I should be able to understand whether the pharmacological action, what I'm going to see, or the efficacy, what I'm going to see, is because of the drug or because of the psychological feeling of the person. Many cases, if you see, the psychology itself will be able to treat many of the conditions in the body. So if they want to, as a part of the clinical study, they want to prove the action is only because of the drug in it, not because of the psychology of it. So what is the difference between a placebo and an active comparator? The objective is to check whether the action is because of the real pharmacological effect. In the active comparator, we just test this against an existing drug. The difference between placebo and active should be very large. between active and the treatment drug should be small. So apart from this, you have the analysis, sample considerations, and much more. The blinding procedures, which I said earlier, a single blind is nothing but only the patient doesn't know whether he's taking drug or a placebo. Double blind, even the patient and the caretaker or the paramedic will not be knowing whether it is drug or the placebo because both of them looks the same. Some cases we use something called as double dummy, where in one group we have test active, reference placebo, and the reverse of it, you have test placebo and reference active. In cases of emergency, for example, the person is being administered with something, the person has serious adverse events. So I need to treat that person, but I don't know what has been administered to the patient, whether I give them placebo or I have given them the drug. In that cases, in the cases of emergency, there is something called as code break. That means you can break the code and see to what patient what has been given so that it will be easy for us to treat the person's adverse conditions. Sometimes you need to compare the combination of products where you can group in such a way that so you have 2 packets to be given to a person. One packet can be active, second can be placebo in one group. Second can be placebo first, followed by active. In the third group, both of them are active. In the fourth group, both of them are placebo. So you can have stratification or combination multiple times. Treatments. So what treatment are you going to give? Whether it is a new treatment which is compared against an established therapy, or the new treatment which is completely checked against the placebo drug, whether it is because of the drug or the psychological things. So how do you compare a treatment? The dosage and administration. So you do the bioavailability and bioequivalency study to check for the new formulation. And the dose is corrected for the patients basing upon the body weight, surface area, severity. I think you read about all these things in your BFARM, Clark's formula, Dilling's formula, right? The dose adjustment is made basing upon that. So you'll be selected for the time of administration, and if the food has any effect, they're going to check. If the person who is enrolling into the study has used any drug in the previous instances, we have to wait till the drug is completely eliminated from the body. We do evaluate the drug interactions and check whether if there is any other drug taken into the person, so you need to advise them. Once the process is continued and the data is obtained or the blood sample is taken or other samples are taken from the patient, the data obtained from these samples is now analyzed. So once the analysis is done, we do check against the hypothesis, whether it is giving any benefit or not. The criteria for different comparability between the groups and how do you treat the analysis and the statistical methods, all of them will be considered. During the process of data analysis, generally, as we said in the clinical trials, it will take multiple years for completion of a clinical trial. So you cannot wait for 3 years or 5 years and then say it is a failure. What we do is we do interim analysis, maybe 6 months once or 1 year once. We do the analysis of the data to check whether the process, what we are going to do is proper or not. So if you are going to do, then the question arises, who will do it? What is the method of it? Whether the blinding code was broken. If the test results are analyzed, who should be knowing the results? And by knowing the results, if it is having any problems or any serious adverse events, whether to continue the trial or to stop the trial, all of them will be considered. The estimation of the sample size, the term sample size refers to the number of patients or subjects in each group or in each clinical trial site. So we need to go and check what is the requirement as per the regulatory authority. Basing on that, we are going to do that. Imperfect data, that means you have got a data, but the data seems to be not perfect. By looking at it, one can identify because of the data or the remarks of it. Sometimes the data is missed. That means there is data which is lost. You able to hear me?
Sir, it is audible.
Okay. In some cases, the data, what you can see is said to be not so proper. So we should be able to understand what information is available, what is recorded, what is the issues related to that. In some of the cases where we say the consent withdrawal happens, that means the subject who has given the consent for participating in the study will now say, I don't want to participate further. He wants to go. And that is the case, the withdrawal or one subject data is gone. In some of the cases where we say the protocol violation happens, that means we say the person should not eat anything after taking the drug, but he will violate by taking the drug. So that is a violation of the protocol. And some cases where you have withdrawal for multiple cases also. So the multicentric clinical trials, if the example for phase 2 and phase 3 trials, which is said to be multicentric, that means you have to do multiple studies or the studies at multiple sites for the same drug and the same protocol. So in that is the case, you have to check what is the number of patients in each center. If there is any slow enrollment in one of the center, how you can substitute that center? You have to plan for investigators meeting, appoint the study coordinators, and many other cases, you need to translate the trial documents into the native languages for the people or the patients to study and understand. You cannot keep everything in English all the time. Publication policy, once the data is obtained, if things are fine, if the results are conducive and better, the report writing committee will start writing the publication, and it can be published as a part of your activity or the outcome. So question comes, who should be getting the authorship, whether investigator, co-investigator, or the sponsor, etc. As a compilation is progressing, we submit the interim report to the regulatory authorities. That is every 6 months or every 1 year, you have to present the data. Please remember, the clinical trial will happen for multiple years, 3 to 7 years, 8 years, the clinical trials will happen. So the data have to be submitted to the regulatory authorities over a period of time. The final report is submitted once everything is over, and the data can be part of your conference presentations or it can be dissertations. So protocol, again, you have a title derived from the aim. You need to have an identification number, version number. That means a version is nothing but when the clinical trial protocol started, it is version 0. Because of some requirements, if you change the contents of the protocol or the process, it becomes version 1. If you make a version 1, that will have the date when it was revised. That becomes the revision date. And when you revise, you have to provide in the summary table what is the basis of revision and what is the new version. To understand things better, we need to give certain flow charts to make things easier. So the golden rules of good clinical practices: write what you want to do. That means you should be very clear how an activity is to be done, which is fulfilling the requirements of good clinical practices, which is a part of your ICH E6 document. So once you have written it properly, then you do as you have written. That means you just follow all the procedures, SOPs, whatever has been defined in your protocol. But is it possible every time? It may not be possible to follow or do things exactly what is written, but sometimes there are certain deviations. So what you do, write what was actually done. So example, you should have taken blood sample at 11:30 a.m. You could not take, you took sample at 11:32 a.m. So write the sample was collected at 11:32 a.m. Don't write it as 11:30, because that 2 minutes will have a lot of consequences for the drug, the what do you call the metabolism or anything you talk of. 2 minutes is a very important time. So these are the 3 important rules. Write what you want to do as you write, more or less. But if you could not do, then write or document what was actually done as a part of your activity. So if something is not written down, that means probably it was not done. So if you have done anything, please write it down or document it. Thank you. So this completes both the presentations of the talk. If you have any questions, you're free to ask.
If you have any doubts, please post in chat box.
Hello, sir.
Yeah, ma'am.
Yes, sir. Session is over, right, sir?
Yeah, madam. Both the presentations are done.
Hello.
Yeah, madam.
Okay, sir. Thank you, sir. So, thank you very much, Dr. Venkatesh, sir, for your nice. So, if students are having any doubts, you can post in chat box. If those are not there, we will wind up the session. Okay, sir. Thank you, sir. Let us
Okay.
meet
Thank you.
later, sir. Okay, thank you.
Thanks a lot. Bye.
Bye, sir.